Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be doing exam 4 review for Biology 2421. Let's look at Chapter 8. This exam will cover Chapters 8, 10, 26, and 27. So let's start with Chapter 8 out of our Brock Biology of Microorganisms textbook, covering viruses and their replication. Again, please study all the slides, know all the material for the exam. Don't just go off of this review. This review is just highlighting some important material, but obviously know all the material for the exam. Now, let's go ahead and get started. Let's see. Obviously, any of these terms, please know these terms. I'm not going to rehash all this material, but know what a capsid is, the difference between naked virus, envelope virus. Remember, an envelope virus does not produce its own envelope, does it? It doesn't make its own membrane. It takes the membrane of a previous host. So it's a host membrane. It might take the plasma membrane of a host. It might take the Golgi membrane of a host. It might take the endoplasmic reticulum membrane of a host. The envelope viruses stole that membrane. All right, so let's know the difference between lytic or virulent infection and lysogenic infection. Remember, lysogeny has two life cycles involved. You can have the lysogenic cycle and you can have the lytic cycle as well. Let's see, the viral genomes. You should know about the viral genomes, how they're quite diverse. They could be DNA, RNA, single strand, double strand. They could be plus strand RNA, minus strand RNA. They could be linear, they could be circular, they could be one chromosome or multiple segments. You should know that the plus strand RNA is the same sense as a mRNA. We should know a little bit about the different types of viruses: bacteriophage, archaeoviruses, animal viruses, plant viruses. You should know what the capsomere is, and it's made up of individual capsid proteins. The capsomeres, I'm sorry, the capsomeres are the individual proteins that make up the capsid. Know that. And so here you could see those capsomeres coming together to form the capsid. You should know the difference between helical and icosahedral, viral symmetry. Let's see, envelope viruses, we touched on that before. And then enzymes of viruses, lysozyme, neuraminidase. RNA replicases. Do you remember the RNA-dependent RNA polymerases? So an RNA-dependent RNA polymerase is a enzyme that copies RNA into complementary RNA. Whereas reverse transcriptase, you should know, reverse transcriptase copies RNA into complementary DNA. And then you should know the life cycle of a virus: attachment, penetration, synthesis, assembly, and release. And you should know the importance of attachment and absorption. Do you remember the story of Timothy Ray Brown, how he got a mutated version of CCR5 in a bone marrow transplant, and that helped him to become immune to HIV, because the HIV virus was no longer able to attach to his cells. Remember that story from class? Again, if you see something in bold, just learn those concepts. Attachment, you should know attachment is very, very important, and it is required for infection. It is required for infection. Then penetration. You should know that with bacteriophage, the capsid stays outside. With bacteriophage, the capsid stays outside. But with animal viruses, usually the capsid comes in as a nucleocapsid. You should know about CRISPR and restriction endonucleases. These are mechanisms by which microorganisms can help to prevent viral infections. Remember, virulent phages, these always kill the host after infection, but temperate phages can switch between lysogeny and lytic conversion. And lysogeny means the genome is either integrated into the bacterial chromosome as a prophage, or it can exist as a plasmid. So here's temperate phage. Remember, temperate phage have 2 life cycles. Temperate phage can enter lysogeny, where there's a prophage, and then that could cycle back in, once induced, it could cycle into the lytic phase as well. What else? And now we're talking about animal viruses. So know the difference between virulent infection, latent infection, persistent infection, transformation. Remember, transformation means that there's a conversion of a normal cell into a tumor cell, into a cancerous cell. You should know about the retroviruses and reverse transcriptase. Very good. So that's good on that front. Let's hop to Chapter 10. Chapter 10 is more about viruses. Let's go ahead and get started with Chapter 10. You should know about roughly the size of genomes of viruses. You should know that RNA genome viruses tend to be smaller than DNA genome viruses. Okay. And you should know a little bit about the Baltimore scheme of viruses. Remember, Class 1, Class 2, Class 3, Class 4, Class 5, Class 6, Class, I think, is there Class 7? Yeah, Class 7 as well. And what's important to know? Well, what's important to know is that all of these viruses need to make an mRNA, right? And from that mRNA, they can produce proteins, right? And they also need to replicate their genomes as well. So all viruses need to replicate their genomes. All viruses need to have some kind of mRNA so that they can make proteins as well. All right, what else did we need to know? Remember the RNA viruses. So real quick, the DNA viruses can use your DNA polymerase to copy the DNA. However, remember the RNA viruses, these are ones that have to use the replicase, you know, the RNA-dependent RNA polymerase or the RNA-dependent DNA polymerase. The RNA viruses need to use their own enzymes to copy their genomes, to copy their information. What else? Oh, you should know that Class 4, these are the single-strand positive RNA viruses. Remember, their genome can be used as mRNA. But you should know that Class 5, these are the negative strand single-strand viruses. Their genomes cannot be used as mRNA. And then you should know with the retroviruses, these require the enzyme reverse transcriptase, which is a RNA-dependent DNA polymerase. All right. You should know about lysis or budding. Viruses can leave the cell via lysis. They break the cell open, or by budding. Usually budding results in, having an enveloped virus, whereas lysis results in not having a naked virus. Usually, not always. Let's see. We talked a little bit about how DNA viruses arose. We should know that DNA is more stable than RNA. DNA is more stable than RNA. We talked about CRISPR. This is good to understand how CRISPR works. CRISPR uses spacers. And remember the mechanism of action of CRISPR. You have viral infection. And once the viral genome is inside the bacterial cell, the Cas proteins, the memorizing complex of Cas proteins, will recognize the PAM sequence, and it will chop out a protospacer next to the PAM sequence. It will then integrate that as a new spacer into the CRISPR gene. And then later on, that sequence can be transcribed outside, transcribed, cleaved, and then that sequence, that spacer, can be conjugated with a Cas protein, and that could be used to cleave any other viral genomes that have the same sequence. So understand how CRISPR works and how, that requires the PAM sequence for identification. It requires cleaving out of a protospacer and integrating that protospacer into the genome between these repeats, these yellow repeats, and then transcription of the genome, cleaving of the genome, and then conjugating the protospacer with Cas proteins and cleaving other like genomes. All right. Then we talked about viroids. We should know the viroids are RNA infectious agents that are completely RNA, small circular single-strand RNA molecules, plant diseases. And then we talked about prions, very important prions. These are infectious proteins, and they cause these diseases here: scrapie, mad cow, Creutzfeldt-Jakob disease, Kuru, chronic wasting disease. And remember how these work. You have everyone has properly folded prion proteins in their neurons. However, exposure to the misfolded prion protein causes a cascade event where all of your properly folded prion proteins also become, they change conformation to the misfolded state, and that causes these aggregates to form, and they causes these plaques to form in the brain, and that causes with a spongy form encephalopathy, this kind of like brain damage that gives the brain a spongy appearance. Awesome. Well, let's move on. Let's keep going strong. Let's go to chapter 26. Chapter 26. Let me change this here. All right, chapter 26. All right, what are we doing here? This is innate immunity. Again, know all the words in bold. Innate immunity does not get better over time. The adaptive immunity does get better over time. It requires specific antigens. Then we have our dendritic cells, neutrophils, macrophages of the innate immune system versus the B cells and the T cells of the adaptive immune system. You should know that all of these are parts of the innate immune system: the skin, the mucus, the lysozyme, flushing, acidity, normal microbiota. All of this stuff is part of the innate immune system. Let's see, what else? We should know about the lymph nodes and the lymphatics. We should know about the bone marrow, the thymus, spleen. All of these are parts of your, lymphoid tissues and your immune system. And then we talked about the lymphatic vessels and lymph capillaries, and then with the blood capillaries, and how there's exchange of white blood cells between the lymphatic capillaries and the circulatory capillaries. We talked about plasma. We talked about serum. Let's see. Leukocytes. You should know the leukocytes means the white blood cells. This includes the lymphocytes, the B cells, and the T cells. The bone marrow and thymus are primary lymphoid organs because this is where the B cells and the T cells mature, respectively. We talked a little bit about dendritic cells, macrophages, neutrophils, mast cells, natural killer cells, T cells, plasma cells. So this is really important. This is something I want you to know, and that is that they're the phagocytic cells of your body. I want you to know which ones they are. The phagocytic cells are the dendritic cells, the monocytes, the macrophages, the neutrophils, and the B cells. All right, those are the phagocytic cells of the body. And then I want you to know which ones are the professional antigen presenting cells. Which ones are the professionals? The professionals are the dendritic cells, the macrophages, and the B cells. Those are the professional antigen presenting cells of the body. Good. Those are the important things right there. Mast cells, remember, what do mast cells do? Mast cells release histamine for vasodilation. They're a type of granulocyte. These neutrophils and mast cells are types of granulocytes. They can release granules. You should know that natural killer cells are involved in the innate immune system, whereas the T cells and the B cells are involved in the adaptive immune system. You should also know about the other granulocytes. So remember, neutrophils, basophils, eosinophils, and mast cells are granulocytes. All right, you should know that the T cells possess T cell receptors and the B cells possess B cell receptors, also known as antibodies or immunoglobulins. We should know what happens during tissue damage. Remember, tissue damage results in cytokine release, chemokine release, resident leukocytes, and damaged cells release cytokines, or chemical mediators that allow communication between white blood cells. Release of cytokines and chemokines draws macrophages and neutrophils to the area as they leave circulation. This is called extravasation or diapedesis. So here's the site of a cut. You should know that tissue damage itself results in acute inflammatory response. So here you can see acute inflammatory response. The neutrophils and the macrophages are squeezing through, doing diapedesis or extravasation, squeezing through the blood vessels to get to the bacterial infection and to phagocytose those bacterial cells. All right, and then we knew how do these phagocytic cells recognize the pathogens? Well, they have pathogen-associated molecular patterns. The pathogens have pathogen-associated molecular patterns, such as peptidoglycan, flagellin, double-strand RNA. These are called PAMPs, and they're recognized by pattern recognition receptors on the white blood cells, on those phagocytic leukocytes. So here you can see the pattern recognition receptor on the phagocyte recognizing those PAMPs, those pathogen-associated molecular patterns on the pathogen. And then remember, once you phagocytose, you bring the substance in as inside of a phagosome, which lyses to a fuses to a lysosome to form the phagolysosome. The phagolysosome introduces hydrolytic enzymes which break down the material, break down the phagocytosed bacteria or virus or whatever, and that breaks down that the biomolecules in that pathogen. And then we talked about inhibiting phagocytosis. Remember, Mycobacteria tuberculosis, it has this waxy mycolic acids, and it's a type of acid-fast cell. So that neutralizes singlet oxygen, and that prevents the phagocytes from destroying the bacterium. Other bacterium can produce leukocidins to cause pus and kill the white blood cells, and also capsules make it difficult for phagocytes to engulf them. So you should know these are three different mechanisms by which phagocytosis can fail due to these virulence factors in the microorganisms. Then we talked about inflammation. Remember the cardinal signs of inflammation: redness, swelling, pain, and heat. All right, what else did we talk about? Localized inflammation. So when you have general inflammation, this can cause fever. Fever is triggered by certain cytokines, specifically interleukin-1, IL-1, which travels to the hypothalamus of the brain, resets the body's temperature setting to a higher level. So You raise your body temperature. Fever-causing cytokines called pyrogens, like IL-1, they increase the body temperature. And why do you want to increase the body temperature? Well, because it increases circulation, so you get more white blood cells to the site of infection. It's also beneficial because some bacteria can't withstand that increased temperature. And lastly, it's important because there's an increase in transferrins in the body, which sequester iron, keeping the iron away from the pathogens and limiting the pathogen growth. So what's systemic shock and systemic inflammation? Septic shock, systemic inflammation, widespread. This is when the infection goes throughout the body, and this can cause multiple organ failure and death due to hypervolemic shock. So your blood pressure goes down, and this can cause multiple organ failure. All right, complement protein. We talked about complement protein. You should know that complement activation occurs one of three ways: classical complement activation, mannose binding lectin activation, and alternative complement activation. Just know of the three ways, but the main one I want you to know is classical complement activation. And this is when an antibody, which is bound to a bacterium or to a pathogen, this is recognized by the complement proteins, and that can lead to activation of the complement proteins. All right, so what do I need you to know about the complement proteins? Again, they're activated one of three ways: either classical, the mannose binding lectin pathway, or the alternative pathway. But once activated, I need you to know these things. Complement protein 3b can serve as an opsonin to help with phagocytosis of a pathogen. Complement protein 5b, 6, 7, 8, and several 9s can insert into the membrane of pathogens like bacteria and form what's called the MAC, membrane attack complex, which will kill the cell due to osmotic shock. And then there are complement proteins C3a and C5a, which can bind to mast cells and promote degranulation of histamine, so this can cause vasodilation. And also C3a and C5a can serve as chemoattractants to help bring neutrophils and other leukocytes to the site of the infection. Then we talked about natural killer cells. You should know that natural killer cells are part of the innate immune system. They have granzymes that they can release that promote apoptosis in a sick cell. They also have perforin, which they can release, which perforates the target membrane. So then you could see here, remember how it works. A normal cell normally expresses MHC1. However, when the cell becomes sick with, it says disease target cell, sick with either viral infection or by becoming transformed into a tumor cell, this can trigger internalization of MHC1 and presentation of a stress protein, and natural killer cells recognize that as a distressed cell. They will then release perforin and granzymes, and that will kill the cell. They will poke holes in the cell and promote apoptosis in the cell. Again, interferons are also another type of innate immune system response. A cell is infected with a virus. That virus replicates inside of the cell, and if the cell recognizes this through a signal transduction event, then the cell can produce what are called interferon proteins that would undergo gene expression of interferon proteins. The interferon will get released from this cell. The interferon will attach to adjacent cells, and what will happen is a signal transduction event in the adjacent cell, which will promote expression of genes that are called antiviral proteins. These antiviral proteins will degrade viral DNA or viral RNA. They will, stop transcription and translation in the cell. They will do a number of things to prevent this cell from being taken over. But one thing I really need you to know is that interferon response does not prevent this cell from being taken over. It will only prevent this cell from, well, actually, hold on. It will not prevent this cell from being infected, okay? The cell will still get infected, but the cell will not become a viral factory. The cell cannot be taken over. Does that make sense? So that's what I want you to know. This adjacent cell, which has received interferon warning, it will still become infected. However, it will not succumb to the virus. It won't be taken over by the virus. All right, so that leads to the end of this chapter 26. Before we hop into the final chapter, let me rest my voice for a minute. We're going to take a quick break time with the Gizmo and Wicket, see what they're up to, and we will be right back to finish off this exam review. Hey everyone, welcome back from break time with Gizmo and Wicket. Let's finish off this exam review with chapter 27, adaptive immunity, also known as specific immunity. Remember, this chapter relies on specificity of either a B cell receptor to an antigen or a T cell receptor to an antigen. And remember, B cell receptors are also known as antibodies. Also remember that each B cell and each T cell has a very specific receptor on its surface that recognizes only one antigen. And the whole point of specific immunity is to have a memory so that subsequent exposures to the same antigen produce a rapid and large reaction, a large immune response, so that you are essentially immune to that disease. Remember what tolerance means? Tolerance, the acquired inability to make an adaptive immune response to one's own antigens. So remember that if you fail tolerance, this results in autoimmunity, response to self, attacking tissues that are your own. And how do the B cells and T cells prevent autoimmunity? They do what's called clonal deletion. Any T cell and any B cell that recognizes self antigens will kill itself, right? That's called clonal deletion. What else? Oh, you should know that B cells and T cells are made in the bone marrow, but the T cells mature in the thymus. That's where the clonal deletion occurs. And the B cells mature in the bone marrow. You see the bone marrow, and that's where clonal deletion occurs. So definitely know that T cells mature in the thymus, B cells mature in the bone marrow. What else did we need to know? Know the difference between an antigen and an epitope. Remember, the epitope is the specific portion of the antigen that is recognized by the antibody. The epitope is the portion of the antigen recognized by a particular antibody. Know the difference between active and passive immune response. Know the difference between natural and artificial immunity. I might ask you examples like this on the exam. What's an example of natural active immunity, natural passive immunity, artificial active, etc., etc.? Know the activation of B cell receptors. Remember, I did a virtual whiteboard on this where, a B cell has a specific receptor or antibody on its surface that recognizes a very specific antigen. It then phagocytoses that antigen. It then breaks up that antigen and presents that antigen on the surface of the cell with MHC2, which is recognized by helper T cells, and the helper T cells will help fully activate the B cells. So know that B cells are not fully activated until they receive help from helper T cells. So know what the helper T cells do, know what the B cells do. So once the B cells become activated, they will proliferate and differentiate into plasma cells that secrete that antibody and memory cells that remember that particular antigen. Then know that antibodies mean immunoglobulins. Know the term opsonization. Remember, opsonization is when a molecule binds to a foreign antigen and makes it easier to engulf by phagocytosis. Know the term neutralization, and antibodies can neutralize toxins or viruses. Know the difference between IgG, IgA, IgM, IgD, IgE. IgG is the most common. You should know IgG is the most common antibody in the body. You should know a little bit about IgM, IgA, IgE, IgD, as you know, is explained on this slide. You should know that IgM is the result of primary antigen response. Remember, primary antigen response results in IgM release, and you should know IgM is a pentamer. You should know about IgA's dimer. Okay. Again, remember primary exposure. So know this stuff: antibody production and structural diversity. You should know what happens during primary antibody response and secondary antibody response. So know that first IgM is released as these pentamers, but then there's a more robust response upon secondary exposure with IgG. So IgG appears upon secondary exposure. IgM appears at a lower level upon initial exposure. Then we talked a little bit about antibodies. So you should know the structure of an antibody. You've got the light chains, two identical light chains and two identical heavy chains to an antibody. Remember, there's the variable region called the CDR region, and then the constant region of the antibody as well. And you should know that antibody diversity is a result of somatic recombination, hypermutation, etc. And you should know that it's the constant region. It's the constant region that dictates the identity of the antibody. So is it IgM? Is it IgG? Is it IgA? Okay. Again, we talked about somatic hypermutation. You should know about major histocompatibility complex. You should know that in humans it's called HLA. You should know MHC class 1 is found on all of your nucleated cells, and you should know that it's recognized by T cells, the cytotoxic T cells, CD8 positive T cells recognize MHC class 1. And also you should know that when a cell has internalized its MHC class 1, that's when the natural killer cells kill the cell. Okay. So MHC class 2, you should know that these are found on the professional antigen presenting cells, dendritic cells, macrophages, B cells. And you should know this too. Remember, all cells, including these professionals, the dendritics, the macrophages, the B cells, they also have MHC class 1. So they have MHC class 1 and class 2. And what does MHC class 2 do on these dendritic cells, macrophages, and B cells? Well, it presents antigen to the helper T cells called the CD4 positive T cells, which are the helper T cells. And those helper T cells help to activate B cells. Okay, and you should know that MHC class 1 is the reason that there are, that tissues might get rejected if you don't have exactly the same MHC or very similar MHC molecules. The immune system will attack that organ when you get a transplant. So polymorphism and polygyny, these cause MHC molecules to look different, right? Polymorphism versus polygyny. What else? We talked about, the difference between what the helper T cells do and what the cytotoxic T cells do. We talked about that. Remember, your professional antigen presenting cells, they will present foreign antigen on MHC2 to helper T cells to activate helper T cells. They will present endogenous antigen on MHC1 to cytotoxic T cells, which can then go do their job and kill the cell. You should know that the T cell receptor recognizes not just antigen, but MHC1 as well. Okay, what else did we want to know? That, yeah, again, know how the cytotoxic T cells work. Remember, a nucleated cell will present antigen on MHC1. If it's a self-antigen, the cytotoxic T cell will do nothing and scoot by. If it's a foreign antigen, the cytotoxic T cell will become activated and release granzymes and perforin to kill the target cell. You don't need to know the difference between helper T cell 1, helper T cell 2, helper T cell 17, or Treg cells. With regard to autoimmunity, you should know that this is when the B cells and the T cells are activated in response to self proteins, and this is what causes tissue damage. And then with regard to superantigens, simply know that proteins capable of eliciting a strong response because they activate more T cells than a normal immune response would. And that is it. We've reached the end of the semester, the end of this exam review. Study these points and know these terms and know these concepts, and I think you'll be fine. It's been awesome. I hope you do well in your last exam, and I'll talk to you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.